Okay, guys, so we'll continue on with antibacterial drugs that target DNA or RNA. So the main class of compounds are the fluoroquinolones. These have really high potency, have a broad spectrum, and if you ingest them, they're readily absorbed from the intestine, so they can get to your bloodstream. Unfortunately, there are some side effects. There are seizures and brain disturbances that can occur with these guys. So the way the fluoroquinolones work is they inhibit the DNA unwinding enzymes and helicases, so thereby stopping the DNA transcription that takes place. So in regards to these medications, there are first, second, third, and fourth generations. The current fourth generation of drugs, the subgroup would be trovafloxacin, and this one is most effective against anaerobic organisms. All right, so the class of drugs that target the cell membranes. One particular class, and the most recognized, are the polymyxins. This is derived naturally from a bacillus strain. One of the unfortunate side effects is it can be toxic to the kidney. Polymyxin is one that is found in Neosporin as well. And of course, that's applied topically, so there usually isn't too much kidney issues with that. So the way the polymyxins work is they interact with the phospholipids in the membrane, and they sort of insert themselves into the phospholipid and disrupt it, which can cause leakage. It's particularly effective in gram-negative bacteria. Polymyxin B and polymyxin E have been used to treat drug-resistant Pseudomonas aeruginosa, as well as urinary tract infections by gram-negative rods. And there is a subgroup of polymyxins, one called daptomycin. This one actually is more effective against the gram-positive bacteria. All right, so we've talked about biofilms before in the class. So biofilms, unfortunately, because there are so many different bacteria species in a biofilm, they're actually much less sensitive to the same antimicrobial drugs. And I think we talked about it when we originally talked about biofilms. The bacteria in biofilms can actually take on different phenotypes when they're expressed at the same time with all these different bacteria. So some of the ways that would be used to treat the biofilm is to interrupt the quorum sensing pathways. Additionally, DNase can sometimes be added to antibiotics. This DNase is going to actually be able to kind of disrupt the clumping of the bacteria together, as well as pre-treatment can be done in some cases. All right, so how do we treat fungal infections? So as you're aware, the fungal infections are caused by eukaryotic organisms. So the antimicrobial agents we were just talking about were all effective against gram-positive, gram-negative bacteria, mycobacterium and such. So here is a list of different agents that have been used to treat fungal infections. The macrolide polymers, amphotericin is a main group which can actually bind to fungal membranes, which are a little bit different sometimes than other eukaryotic membranes. The azoles, so these can interfere with sterol synthesis in fungi, and in particular, there's some specific ones that are listed here that can be used for different types of fungal infections. So those would be the main two fungal drugs that you should be aware of. All right, so what about protozoans and helminths? Okay, so again, the same idea as with the fungus, different types of microorganisms that are eukaryotic in origin. So there has been a drug that's been in place for malaria, which is caused by a protozoan called quinine. So this has actually been the principal treatment for hundreds of years. In recent years, it has been replaced by derivatives of quinine, chloroquine and primaquine, because they are less toxic. As well, there is another drug for other types of protozoan infections, especially if they're caused by an amoeba. So metronidazole is an effective amoebicide. When we get into infections that are caused by helminths, here is a list of some of the drugs that have been used and effectively against those helminth infections. So we also have a class of agents that are effective against viruses. Viruses, when we think about the chemicals that affect them, there's three major modes of actions. One way is that we can actually penetrate the virus from doing the penetration step of getting into the host cell. The second one would be to actually block transcription or translation of the viral molecules so that they can't replicate and then multiply in the host cell. And then the last step is to actually prevent what's known as budding, where we actually prevent the mature viral particles from being released from the host cell. All right, so the first mode of action we talked about is inhibition of viral entry. So in this case, there's a couple of classes. The first one, is an effective one against blocking HIV infection. So there are these receptors, GP41, on the cell receptor, and they actually prevent that interaction between the GP41 viral receptors and the cell receptors on the host cell. Influenza virus also can be inhibited, and in this case, influenza actually will fuse with the cell membrane, but if certain antiviral drugs are used, it can actually stop an enzyme in influenza called neuraminidase, which is required for entry of the viral particles into the cell. All right, the second class of ways that viruses can be inhibited in cells is through inhibition of the nucleic acid synthesis. So in number 4 here, which is for acyclovir, in this particular case, there are some purine analogs. So a purine analog is something that mimics the purine nucleotide, and it's going to terminate DNA replication. This has been very effective for the herpes virus. Ribavirin works in the same way, but this is more specific for RSV and some of the hemorrhagic fever viruses, such as Ebola. There are some mechanisms that actually can block viral production by using reverse transcriptase inhibitors. This has been effective in HIV, as well as using non-nucleotide analog reverse transcriptase inhibitors In this case, they actually bind to the HIV reverse transcriptase site, stopping its action. And then the last class is going to be for inhibition of viral assembly and release. So in this particular case, and again, these are antivirals that have been effective against HIV. These types of drugs are sometimes just referred to as protease inhibitors. In this case, they insert into the HIV protease and they stop its action and essentially stop the release of the infectious particles. All right, we'll continue on to section 12.4. This is talking all about antimicrobial resistance, which we hear about all the time these days. So, in regards to drug resistance, essentially, drug resistance can be a response naturally by the microorganisms. It's thought to be an adaptive response that allows the microorganism to start tolerating a drug that would ordinarily, kill it or be inhibitory. So it's thought that just because of the genetic versatility and the fact that there's so many microbes out there that they can, over time, adapt to particular drugs that might be in the environment. So there are really two types of resistance that can occur. Intrinsic would be where bacteria must be resistant to an antibiotic that they themselves produce. This kind of makes sense because if they're going to produce that antibiotic, it shouldn't kill them. So the intrinsic is there, it's a natural response so that they're able to produce this antibiotic but not kill themselves in the process. Acquired is the one that's been, something that takes time. So this is where the bacterial resistance to the drug takes over in a population, but previously the microbe would have been sensitive or killed off by that drug. So how does drug resistance develop? It's thought that this resistance is really just due to spontaneous mutation. So in this case, there's minimal chance that the mutation will actually be advantageous, and even a smaller chance that the mutation is actually going to give rise to drug resistance. But the microbial population is so large, if you think about it, even in our bodies, we have 10 times more microbes than we do human cells. So it's thought this large population and the fact that there is a constant rate of mutation, even if it's very small, ensures that a positive mutation that would confer resistance do actually occur over time. So again, how does this drug resistance develop? This is usually through a process of horizontal transfer through resistance factors or R factors. We did not get into these processes very much, but bacteria are able to transfer their plasmids through 3 different processes: conjugation, transformation, or transduction. So these are the 3 sort of methods by which the plasmids can be transferred. As well, there are what are known as transposons. This is where a duplicated and inserted sequence goes from one plasmid to another, and then it goes actually onto the chromosome as well. So instead of just transferring plasmids, with the transposons, the plasmid gene sequence gets inserted into the chromosome.